Food and oxygen are non-negotiable when it comes to life. If not for them, most organisms will not be able to survive. And both of those are gifts that we get from plants, because plants perform this remarkable biological process called photosynthesis, as a product of which we get food and oxygen. And in this chapter called Photosynthesis in Higher Plants, we are going to learn about the chemistry of what really happens inside of plant cells when they perform photosynthesis. This is a high-weighted chapter for your NEET, and if you want class notes along with 3PP questions to solve, you can access this free batch on the PW app called Complete Botany by Shalini Ma'am. The link to this batch is provided to you in the description. So click away, explore our app. There are some amazing features that are going to help you get one step closer to your dream of becoming a doctor. Now, without any further delay, let's get today's session started. Since this is the first chapter in the unit Plant Physiology, we'll start off with the unit introduction. Before this, we've already discussed diversity unit, we've discussed cell unit, and also structural organization. So in this unit's introduction, we briefly touch upon what we have learnt earlier. So it begins like this: the description of structure and variation. Structure and variation of living organisms, which we have done in the past in the other units. Eventually, what happened was, when scientists started to describe the structure of living organisms, started to look at how they are all so different from each other, started to look at the variation. Apparently, there were two irreconcilable perspectives on biology. See, when we look at biology as a field or as a branch of science, within biology, you have many levels of organization. Okay, so levels of organization you already know. You've learnt about that in animal kingdom for classification, which one has a loose cell aggregates type, which one has tissue level of organization, which one has organ level of organization. All about this you've learnt. They are some levels of organization. Apart from what you've learnt in animal kingdom, there are few more levels of organization in biology. What they are, I'll tell you right now. So we start off with what are called macromolecules. You know what macromolecules are, right? You would have learnt about that in biomolecules. So macromolecules include your carbohydrates, proteins, lipids, nucleic acids, and all of that. All of these macromolecules that are found in living organisms, they come together to form cell organelles. Different cell organelles come together to form cells. Cells form tissues, tissues form organs, organs form organ systems, and so on. And organ systems will form an organism. This is how much you know so far. Further, beyond the organism level, what do we have? We have what is called a population next. Population is a group of individuals belonging to the same species, occupying a particular area at a particular time. That's what a population is. Next, we have community. Many populations coming together is called a community. Next, we have ecosystem. Ecosystem is many communities along with their abiotic components, which includes water, soil, sunlight, wind, air, whatever else. Okay, so that is the ecosystem. And then you have large, huge geographical areas called biomes, which are like huge ecosystems with their characteristic flora, fauna, and even climatic conditions. And then you have biosphere, which essentially is the entire living organisms and all of the biomes present on our planet. Okay, so these are the levels of organization you can find in biology. So what they're trying to say here is, beyond the organism level and above, which means organism, population, community, ecosystem, biome, biosphere, all of these together will be studied under ecology. Okay, so one is described at organism level and above. That is our ecology. The second branch is cellular and molecular level of organisms. So here we are looking inside of cells. How are cells able to function the way they do? What exactly is happening during biological processes? How exactly are cells performing photosynthesis or respiration or protein synthesis or whatever that is? So what exactly at the molecular level is happening? How is one chemical reacting with another chemical? How is one molecule reacting with another molecule? So you're looking at this not just at the cellular level, you're looking at it inside the cell at the molecular level. So this is called physiology and biochemistry. Okay, so in physiology and biochemistry, that's what you will learn about. Okay, so description of physiological processes in flowering plants is an example for this. So in this unit, that is what we're going to learn about. Firstly, we'll start off with photosynthesis, this chapter, following which we will discuss respiration and then plant growth and development. Okay, so plant growth and development, you will learn about what are the various factors that regulate the growth and development in plants and all of that. Okay, so are described in this plant physiology unit. So let's get started. Also, since this is the first chapter in the unit, we have to learn about a scientist. Here we're going to learn about a person called Melvin Calvin. He was born in the year 1911 in April in a place called Minnesota. He received his PhD in chemistry from the University of Minnesota, following which he served as a professor of chemistry at the University of California. After World War II, at around the time World War II, you know how Hiroshima, Nagasaki bombings and the Pearl Harbor in America getting destroyed, all of that were happening. People were putting radioactivity to very wrong use. But Melvin Calvin, he used the same radioactivity to kind of find out how exactly the process of carbon fixation takes place in plants. Okay, so he, along with another scientist called J.A. Basham, he studied reactions in green plants forming sugar, that is nothing but our photosynthesis, and other substances from raw materials like carbon dioxide, water and minerals by labeling the carbon dioxide with C14, which is a radioactive isotope of carbon, right? So Calvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigment molecules and other substances. The mapping of this pathway of carbon assimilation won him the Nobel Prize in the year 1961. So, even today, some of the principles that he suggested are being used. So, that's about Calvin's contribution, Mr. Calvin's contribution. Yeah. So today we know that photosynthesis takes in carbon dioxide from the atmosphere, it synthesizes glucose, releases oxygen as a byproduct. We know everything about how, pretty much everything about how photosynthesis takes place. But back in the day, people did not know a lot about photosynthesis. For us to know whatever we know today, many people have performed many experiments, and as a result of their research, we have the knowledge about photosynthesis today. So first, let's start off by talking about some experiments people have performed in the past, which has led us to gaining knowledge about photosynthesis today. The first experiment that we'll be talking about today is the variegated leaf experiment. First of all, what is a variegated leaf? It's a fancy term for a leaf that has more than one color. Obviously, it will have green color. In most cases, green will be one of the colors. Along with green, if there's any other color, you call it a variegated leaf. They're very pretty to look at. Actually, the reason why some of the plants have evolved to produce variegated leaves is because most animals think that if there is many patchy colors or a different colored leaf, the animals will assume that the plant is infected and might not feed on it. So this is kind of a defense mechanism for those plants. Okay, so yeah, very pretty to look at, and it's grown as ornamentals in most homes and restaurants and everywhere, right? So these are variegated leaves. So in this variegated leaf experiment, what we do is we test for the presence of starch. Why starch? What does starch have to do with photosynthesis? What is photosynthesis? Carbon dioxide utilized to synthesize glucose. Glucose will not be retained as glucose within the plant cells. Glucose, if it has to get transported, will get converted to sucrose. If it has to be, stored within the plant cells, it will get converted to starch. So if the plant is making glucose and it's storing it, then it will be in the form of starch. If the plant is showing the presence of starch, that means photosynthesis has happened, glucose has been synthesized. Okay, so you're not directly testing for glucose, but for starch to test for whether photosynthesis is happening or not. Now, this is the leaf. How do you test for the presence of starch? There's something called a starch test where we use iodine. Iodine solution, it will appear somewhat honey-like in color. Okay, so it's somewhat yellowish, golden, brown, whatever you call that color, it's like that. So whenever you add it on a sample, and if the sample changes color from its original color to bluish black, that indicates that the sample has starch present in it. That color change you will see only if the starch is present in that sample. If starch is not there, it will retain its original color. So when a leaf from a variegated plant, a variegated leaf from a plant is taken out and you add iodine solution on top of it, after a while you will notice how only in the green parts there will be a color change to bluish black, but the remaining central other color part will remain as it is. It will show the same color as iodine. So this indicates that green color, starch is present only in the green regions of the plant. That kind of indicates that green color is essential for photosynthesis. Okay, this is our first experiment. What is our second experiment? It's called Moll's half-leaf experiment. Here what they do is they take another potted plant. One of the leaves of the potted plant is inserted into a bottle. This bottle has a cork that has a slit. Through the slit, half of the leaf is inserted into a bottle, and it's not like an empty bottle. Within the bottle, you will either have a cotton that is soaked with KOH solution, or you will have KOH solution as it is. This KOH, what does it do? It will absorb whatever carbon dioxide is present inside that bottle. So keep this entire setup in the sun for a while, and then you will check for the presence of starch again using the same starch test using iodine. So when you take this one leaf and add iodine solution on top of it, you will notice how only the part that was exposed outside turns bluish black, and the remaining part that was present inside the bottle, that will remain green in color, no change over there. So this indicates that starch synthesis has happened only here, photosynthesis has taken place only here. Okay, so also you should remember, you might think maybe yesterday the plant would have photosynthesized, so some amount of starch would be stored even over here. This should also turn green. Before this experiment is performed, they will keep the plant in the dark for some time to ensure that without photosynthesis, see, just like how when we don't eat food the next morning we don't die, right? Because there sometimes we would have eaten so many extra calories that it would have gotten stored in our body in the form of fat or whatever else. So when you're starving or when you're fasting, then whatever stored reserve food is there in our body, that will start getting broken down and used for energy. Same thing happens even in plants. Starch is the reserve food there. If the plant is not exposed to sunlight, newer glucose is not being synthesized, existing starch will get used up. So for a period of time, maybe for a couple of days, 1-2 days, the plant will be kept completely in darkness so that it utilizes whatever starch it already has. Once you're sure that it has exhausted all of the starch, you take this plant and keep it in the sunlight and do the setup, and later check for whether starch is present or not. Okay, so then you'll get to know that this has not produced any glucose, photosynthesis has not taken place. Why? What is the difference between this half of the leaf and this half? This was exposed to the surrounding air which had carbon dioxide, while this wasn't. So that's why it did not perform photosynthesis. So similar to how we found out green color is essential for photosynthesis, now we know that carbon dioxide is also absolutely essential. Next we have Joseph Priestley's experiment, one of the earliest experiments that gave us little insights into what photosynthesis is as a process. So this Joseph Priestley, priest means what? Priest is like pujari, right? So priest uses a bell in temples. So Joseph Priestley performed the bell jar experiment. Remember it like that. So what did he use for his experiment? He used candles, he used mice, and he used mint plant, okay? And a bell jar, of course. So what did he do first? He lights up a candle, covers that using a bell jar, and he will notice that after a period of time the light, the candle will get extinguished, like the light will go off, and later he will place a mouse inside the bell jar. After a while the mouse will also die. So he believes that a burning candle and a mouse breathing will take away something from the air. Okay, so they will damage the air and take away something from the air. What he does next is, within the bell jar, he will place a candle, he will place a mint plant and a mouse together. So for as long as the mint plant is present inside the bell jar, the candle also will not get extinguished and the mouse will also not die. So he says, whatever the burning candle and the breathing mouse is taking away from the air, damaging the air, that thing is given back to the air by a plant. Okay, by a plant. That is why these burning continues and the animal continues to survive. Like, right, for burning also oxygen is essential for combustion, right? And for breathing, of course, we need oxygen. Mouse is an aerobic animal, so it also requires oxygen. In the absence of oxygen, it cannot survive. Now, back then when he performed this experiment, right at that time, he did not realize that what was taken away from the air, what was given back to the air was oxygen. Much later, he himself in 1774, I suppose, he discovered that thing to be oxygen. So we know from this experiment that plant performs a process, we don't know what it is exactly, but that liberates oxygen, that releases oxygen is something that we figured from Joseph Priestley's bell jar experiment. Next we have Jan Ingenhouse's experiment. Ingenhouse is his name, I don't know, it sounds very weird. His name has a house. Look at this setup, this looks like a house, doesn't it? Please say yes. Yeah, so that looks like a house to me. So that's how I will remember this experimental setup with what this guy did. So he, around the same time in the 1700s, he performed an experiment that showed sunlight is essential for photosynthesis. His experimental setup is very, very simple. You have like a beaker containing water. Inside that you have, what is this called, like a conical flask sort of a thing, or it is called a funnel, right? That sort of a thing. Inside that you have an aquatic plant, in this case a hydrilla. On top of that you will invert a test tube. He notices that every time this setup is kept in the sunlight, there will be tiny, tiny bubbles that appear on top of the plant. They will also get accumulated at the tip of the test tube, or at the base in this case of this particular test tube. And when checked later, they will identify that it is oxygen. But when they place the exact same setup in darkness, oxygen will not get produced. Those bubbles will not form. You can test this also. Take any leaf that you have growing in and around your house, keep it in a plate or a glass or whatever, pour some water into it and keep it in the sunlight. 20 minutes later, if you go outside, you will be able to see those oxygen bubbles. Okay. So yeah, that happens. So by this, we got to know that sunlight is absolutely essential for the process using which plants release oxygen. If not for sunlight, oxygen will not get produced. So from this, we know that sunlight is essential. Now, moving on. There's another person called Julius von Sachs, much later in the 1800s. He discovered that glucose will get produced when plants grow. When plants are growing, from being short to becoming tall as they are growing in size, they synthesize glucose is something he provided evidence for. What exactly he did in his experiments is mentioned nowhere, so don't worry about it. So that's what he said. What he also said is, in plants, there are special bodies. Okay, today we know them as chloroplasts, but back then we didn't know what chloroplasts are. He said that in plants, especially in the leaves, there are special bodies, and in those special bodies there is a green substance. Today we know that it is the pigment chlorophyll. Okay, so it is within these that photosynthesis happens, is what he suggested. And like I mentioned earlier, glucose is made in the green parts of the plant, and in plants it is stored as starch. So that was the contribution of Julius von Sachs. Sachs kind of rhymes or reminds you of starch, so you can remember. Okay, so SACH, it's only missing like T and R. So that's how I remember it. So that was his experiment. Next. Yeah, T.W. Engelmann's experiment. Angle sounds similar, right? So here you have a triangle. A triangle has these angles. So that's why it's called, that's not why it's called Engelmann's experiment. That is how you can remember what Engelmann's experiment is about. Okay, angle, prism, Engelmann. Well, he wanted to know if all the wavelengths of the visible spectrum were equally effective in photosynthesis. He wanted to know if the plants would respond the same way to all the wavelengths of light, basically. So what he did was, he took a filamentous green alga called Cladophora. Yeah, here you are. Okay, so filamentous means you know, right? So that alga appears like this, but if you take a section of it and put it under the microscope, it has cells lying on top of each other forming a filament of sorts. So he took this filamentous alga called Cladophora. He was growing it in a medium, of course, and then he let this split light incident on Cladophora such that different regions of this Cladophora filament received different wavelengths of light. Now, within the same suspension where he was growing this Cladophora, he had an aerobic bacterium. Okay, so this aerobic bacterium had flagella, so it was motile. You know, aerobic bacteria obviously require oxygen for surviving. So wherever more oxygen was available, they would go and just, settle there. So here, when he exposed this Cladophora filament to different wavelengths of light, 400 to 700 nanometer, and within the same suspension, he had aerobic bacteria, which are represented by these kuti black dots. He noticed that most of these bacteria started to accumulate near the red and the blue regions, indicating that is where more oxygen evolution is happening. Where is oxygen coming from? In Cladophora. Cladophora is an alga, it is capable of photosynthesis, it is going to evolve oxygen as a byproduct. So wherever more photosynthesis is happening, over there more oxygen is getting evolved. So wherever more oxygen is there, these guys are going there because that is more suitable for them to grow. So with this, we kind of figured that Not all wavelengths of light are equally effective with respect to photosynthesis. Um, Blue and red are more preferred by plants. That is when more photosynthesis happens, is what we got to know. Now, because of all of these experiments, by the mid 19th century, which is 1850 something, mid 19th century, this was the empirical equation of photosynthesis. People knew carbon dioxide is required, water is required. It happens in the presence of sunlight, and green color substances called chlorophyll, and eventually glucose would get produced and oxygen would get released. This is what we got to know. Much later, another researcher called Cornelius van Niel, he started studying photosynthesis not in eukaryotic organisms but in bacteria, a type of bacteria called green sulfur bacteria and purple sulfur bacteria. In these bacteria, when photosynthesis happened, oxygen would not evolve. Such a type of photosynthesis is called anoxygenic photosynthesis. Anoxygenic photosynthesis. Okay, so here in anoxygenic photosynthesis, oxygen would not get evolved. Usually when we say plants take in carbon dioxide and give out oxygen during photosynthesis, we tend to think that carbon dioxide has an oxygen, right? Carbon, C, one carbon atom, two oxygen atoms is what makes one carbon dioxide molecule. So in carbon dioxide also there is oxygen, in water also there is oxygen. Since we always say plants take in carbon dioxide and give out oxygen, it's easy to assume that the oxygen present in carbon dioxide is what will get released as molecular oxygen, right? Because when we say plants take in carbon dioxide and give out oxygen, we don't talk about water. We're talking essentially about carbon dioxide and oxygen, which are both gases. But from his experiments, we got to know that the oxygen present in water is what will get released as oxygen from plants. How? Because in plants, because in bacteria that he worked with, which is green and purple sulfur bacteria, in those bacteria, the hydrogen donor wasn't water. Instead, they made use of hydrogen sulfide as a hydrogen donor, and in such plants, oxygen was not evolving. Instead, yeah, sulfur was getting produced as one of the products. So, this kind of helped us understand that the oxygen that is evolved by green plants comes from water and not from carbon dioxide. Later, this was even confirmed by using radioactive isotopes, that the oxygen that is released is not from carbon dioxide but from water. Okay. Now, this is a table of all of the experiments that we've learnt about so far so that you get everything in one place, easier for your revision. Okay. Now, we'll speak of the overall equation of photosynthesis that we know today. In photosynthesis, carbon dioxide and water are raw materials. As products, glucose and oxygen are produced, and in this process, light and chlorophyll or even other pigments are absolutely essential. Photosynthesis is a redox reaction where water is getting oxidized to oxygen, carbon dioxide is getting reduced to form glucose. Okay, so this is how it is. Though we usually represent photosynthesis as a single step reaction like this, it's not that easy. Photosynthesis happens in two stages. Think about it like that, like this. Okay. So whenever you go to this pani puri shops and all of that, you don't have like pani puri made in one go. It's not like a one pot recipe, right? To make that pani puri, you need that puri, you need that filling inside, and you need that flavored pani and all of that. So first, for preparation, during preparation, you take and make and fry your puris and keep it ready. You make that potato preparation ready and you make your flavored pani ready. All of that is ready. And then later you assemble it and you get that whole pani puri, right? Something very similar happens in photosynthesis too. I don't know, that might be like a super simplified analogy, maybe not very accurate, but just to give you an idea, I gave you that example. That's what I could think of right now. And here what happens is an anabolic process. During photosynthesis, a very small Kutti molecule, carbon dioxide, which contains just one carbon, 6 such molecules are going to combine to form one big molecule which contains 6 carbons. So it's an anabolic reaction. Whenever anything is getting built inside a cell, energy will be consumed. Plants for this reaction cannot directly use sun's energy. Anything that has to happen inside a cell, energy is utilized in the form of ATP only. So first, what do we need? For this to happen, energy is required. That energy plants are obtaining from sunlight, but they can't directly use that sunlight. So that absorbed energy from sunlight has to first be converted into ATP. So in the first preparatory phase, what they do is they trap. They here is pigments. Pigments in leaves, it will absorb different wavelengths of sunlight. That energy is used to produce ATP. So you're absorbing sun's energy, converting that to ATP, keeping ATP ready. And that ATP that is produced by trapping the sun's energy is used later here to produce glucose from carbon dioxide. So the first phase where sunlight is being trapped, captured by pigments, and that energy is converted to ATP, that is called the light dependent phase or the light reaction. Okay. So during light reaction, obviously sunlight will be involved. If sunlight has to be trapped, any molecule cannot absorb sunlight. Specific pigment molecules are responsible for absorbing sunlight. So pigments are involved. So the sunlight is trapped by pigments such as chlorophyll, not chlorophyll alone. Many other pigments are there, we'll talk about it. So chlorophyll absorbs the sunlight. Using that energy, high energy molecules such as ATP will get produced. Also another molecule called NADPH will get produced. NADPH is short for nicotinamide adenine dinucleotide phosphate. H just represents that it's in reduced form. Okay. See, so this happens. And while light reaction is happening, water molecule will split to give oxygen, protons and electrons. Okay, so this is what happens during the light reaction. The splitting of water is called photolysis of water. Usually water is a very stable compound. It does not break off like that very easily. But here during photosynthesis, in the presence of sunlight and with the help of certain enzymes and water splitting complex and all of that, water will split. Okay, so during the light reaction, you can see that water splits to form oxygen. Sunlight is used, trapped by chlorophyll to produce energy. Carbon dioxide is not yet touched upon. Glucose is not yet formed. Carbon dioxide and glucose have no role in light reaction. What happens in the dark reaction? Dark reaction is also called light independent phase or biosynthetic phase. So during the dark reaction, carbon dioxide will make use of ATP and NADPH and produce glucose. Okay, so in this process, light is not directly required, but the products of light reaction are required. This process does not necessarily happen in the dark. It's not like just because it's called dark reaction during the day, it will not happen. It's not like that. It's called dark reaction. It's kind of like a misnomer. This process does not happen in the dark. It happens simultaneously along with light reaction. It's called dark reaction, just like an opposite for light reaction, because it does not directly depend on sunlight, but it depends on the products of the light reaction. Okay, so that's why it is called the dark reaction. Now, see, when photosynthesis happens, you have plants, right? Plants have many different leaves. In leaves, mesophyll cells are present. Within the mesophyll cells, chloroplasts are present. Within the chloroplasts, chlorophyll, pigment and other pigments are present. So, when photosynthesis happens, light is incident on the entire plant. It is incident on the leaves too. If you look at the anatomy of the leaves epidermal cells except for guard cells does not contain chloroplast. So, within the epidermal cell layer, mostly photosynthesis will not take place. Major site where photosynthesis happens is within the mesophyll cells. Why so? Because mesophyll cells are laid and it is filled with chloroplasts. So if you take one mesophyll cell, we don't need to worry about the other parts as of now. Cell wall is there, cell membrane is there, vacuole, nucleus, other organelles, all of those are there. But what is relevant to us right now is the chloroplast. So it will contain chloroplasts. If you take a look at the chloroplast has an outer membrane, it has an inner membrane. Within that there is stroma. Within the stroma there are thylakoid membranes, and these thylakoid membranes, these thylakoids stack on top of each other to form what is called the grana. Now, if you look at the thylakoid membranes, they are very similar in structure to the membranes of the plasma membrane. I've told you, every time I say membrane, it could be of nucleus, chloroplast, mitochondria, Golgi. Every time we're talking about membranes, we're talking about the unit membrane that is very similar in structure to the phospholipid bilayer. Okay, so what happens here? This is the thylakoid. Thylakoid is not like a flat solid thing. It has a membrane that is made up of lipids, so it has like that oil like consistency, remember I told you, and then within that there is space. The space within the thylakoid is called a lumen. How do I show thylakoid? I have this thing here, this dabba. It's not going to be solid like a dabba, but just for your visualization I will show it to you. So this is one thylakoid, let's say. This thylakoid is sitting inside the stroma. It's located inside the stroma. Inside the thylakoid there is space. Okay, so that space is what we refer to as the lumen. So if you look at this membrane, this is in the stroma. So outside this membrane, stroma is there. Inside this membrane, what is there? The lumen is there. Lumen is not empty space. It also consists of a fluid that is similar to the stroma. Okay, a fluid, certain enzymes and all of that will be present. So this is the membrane that we are talking about now. So if you look at this membrane, it is similar in structure to the plasma membrane. In the plasma membrane, remember we've learned about these transmembrane proteins, extrinsic proteins, integral proteins and all of that. Similar to how you have proteins present in the plasma membrane, you have certain pigment complexes present in the thylakoid membranes. Okay, so when I say chloroplast contains chlorophyll, where do you visualize chlorophyll to be present? Is it floating inside the stroma? Is it dissolved in the stroma? Where is it located exactly? The pigments that are involved in photosynthesis are located in the thylakoid membranes. Not only in the thylakoid membranes, they are present in the stroma lamellae too. Okay, so and these pigments are not present individually. Hundreds of them come together, form a complex, and they are located here. Okay, so this is called a photosystem. More about that I'll tell you. Okay, so within each photosystem, you will find hundreds of pigments, of which chlorophyll A is one type of pigment. You have chlorophyll A, chlorophyll B, carotenes, xanthophylls, all of them will be present in different numbers in the photosystem. Along with that, certain enzymes, proteins, electron carriers will also be present. That is called a photosystem. So it is here that the light dependent phase will take place. It is here that the light will be trapped and energy will be produced. See, if this is the thylakoid membrane, I'm sorry, this is the thylakoid, let's say, this is located inside the stroma. In this membrane, light reaction is happening. So when light reaction happens, ATP will get produced. The ATP that is getting produced will get released here in the stroma. Using that ATP, dark reaction or glucosynthesis will happen inside the stroma. So light reactions site is the thylakoid membrane, stroma lamellae. Dark reaction site is in the stroma. That is where ATP, see, this membrane will produce ATP and release it outside. That is made use of by the chloroplast in the dark reaction in the stroma present around it. Okay, yeah, so that is that. That is the site of photosynthesis. Light reaction happens in the thylakoid membrane while dark reaction takes place in the stroma. Moving on. Depending upon what is the intensity of light available to the plant, the chloroplast will align itself differently inside the mesophyll cells. If the light is of low intensity, very weak light is incident on the plant will want to utilize that light to its best ability to photosynthesize. So the leaves will, the chloroplast within the leaves will start appearing. See, if this is the mesophyll cell, you're looking at one mesophyll cell here. This is towards the upper epidermis, you can assume. So if there is less light there, you want to maximize how much how much chloroplast is, how much light is available to the plant so that you can maximize photosynthesis. So in that case, again, we'll take our little dabba. So if this is our cell, let's say longitudinally, this is where it is present. All of the chloroplasts will come and align here. Okay, so if you look at it from above, they are fully covering this top surface so that maximum light will incident and they can photosynthesize. If the if the light intensity is too harsh, maybe during midday where light is super intense, it can damage the pigments if it's exposed to high light intensities for very long period of time. At that time, they will start occupying these sides. Okay, so that they can kind of reduce the damage. So if you look at it from above, they will all be located towards the side. Yeah. See how clever plants are? Beauties. Yeah. So this is the structure of chloroplast, a real micrograph, and here you can see these are the thylakoid membranes. Thylakoids basically they stacked on top of each other. The entire stack, like I've already told you, is called the granum. All of these colored candy-like structures that you're looking at here, they are our complexes. It could contain pigments, it could contain proteins. They are involved in photosynthesis. So that is what is represented here. Okay. Now, I told you that in photosynthesis, chlorophyll is not the only pigment. There are many other pigments that are involved. Which all pigments can you find in a leaf? If you have to find out, we have to perform something called a paper chromatography, where you take the leaf, you grind it in a mixture of, I think, petroleum ether, acetone and ethanol. You make a mixture of sorts and then you grind your leaf with that mixture. What happens is the leaves that have the leaves that have these pigments, they are present inside the lipid membrane. It's located here, right? The pigments are here. They do not dissolve in water. So even if you grind in water, your pigments will not dissolve in water. So that's why we're using organic solvent mixture. When you do that, these pigments will dissolve in that. Then what do? You strain it. You strain it, you filter it, you take a little bit of this, one drop of it on a capillary tube, and then you blot it on what is called a Whatman paper, I think this is called, or a chromatography sheet. So you place it here, and the same mixture of organic solvent that you use to crush and grind the leaves, you use that here, put it in a beaker, just so that this much is covered. Okay, so it's just touching that liquid there. So here it will dissolve in that and it will start separating out. And once it starts separating, you can see many different pigments that are separated. Of course, you will have chlorophyll A, chlorophyll B, carotenes, xanthophylls. Chlorophyll A appears somewhat bluish green to bright green in color, and chlorophyll B appears yellowish green. Carotenes are yellowish to orange, xanthophylls are yellow. Okay, so these are the pigments that you can find in plants. Now, all of these pigments are involved in photosynthesis indirectly, but primarily, most importantly, the pigment that is involved is chlorophyll A. If you remember when we discussed plant kingdom, I told you red algae, green algae, brown algae, all of them have chlorophyll A in common. In addition to that, other pigments are present, right? So, chlorophyll A is absolutely essential for photosynthesis. That is where the reaction center is. I mean, that's what is present at the reaction center, and it is directly involved in the process of photosynthesis. Along with that, other pigments also will be present. So, how is chlorophyll A different from chlorophyll B or C or whatever? If you are interested, I can tell you the difference between chlorophyll A and chlorophyll B structurally. Both of them will have porphyrin head and it will have a long hydrocarbon tail. In the porphyrin head, there is this region where there is an R group. Just because of a small difference in R group, you have two different types of chlorophylls. Chlorophyll A has CH3, methyl, right? That as the R group, and chlorophyll B has an aldehyde group. Okay, so that is how the two chlorophylls are different from each other. Now, there's something called the absorption spectrum. We'll talk about that. Absorption spectrum is something that you plot in a graph using the absorbance values of different pigments. I told you how the mixture of pigments that you find in leaves can be separated using paper chromatography, right? So from that, let's say you have isolated chlorophyll A. You want to find out whether chlorophyll A will absorb the same amount of light across the visible spectrum. So what do? There is an instrument called a spectrophotometer. You will place your sample inside the spectrophotometer here. There will be a small square shaped tube of sorts that is called a cuvette. Inside that you will add your chlorophyll A separated pigment, just chlorophyll A you will add, not that mixture of pigments. Once you've added it, within the spectrophotometer you can set what wavelength of light you want the pigment to be incident with. So this instrument kind of measures what is the incident wavelength, what is getting transmitted from that pigment, and it will kind of calculate and give you what is called an absorbance value. It absorbs some and it emits some, right? So based on that difference, it will give you an absorbance value. So you will calculate the absorbance value for chlorophyll at 400 nanometer, 410 nanometer, 420 nanometer, and every wavelength that is possible. And you will plot it on a graph where you have wavelength against the x-axis and absorbance value on the y-axis. So you will notice how this is the end. Yeah. So this is the graph that we got for chlorophyll A. You can see how at 400, here's how much it has absorbed. Okay. So at maybe this is 410 or something, at 400 here's how much it has absorbed, at 500 here's how much, and then at around 700, maybe 650, 680 it has absorbed this much. So you know that it absorbs highest maximal wavelength is near 400 and 700 nanometer, right? And then similarly you do something, you do something similar for chlorophyll B, and then you do something similar for carotenoids as well, and you get this absorption spectrum. This is called the absorption spectrum. Very clearly I want you to notice how at the x-axis you have various wavelengths, and at the y-axis you have the absorbance of pigments. Okay. That is called the absorption spectrum. So there's something else called the action spectrum. In action spectrum, you do not separate out pigments, okay? So you check for the rate of photosynthesis. How can you check the rate of photosynthesis? Based on how much product is being produced with respect to time, right? So what are the products of photosynthesis? Glucose is the primary product. As a byproduct, oxygen is also produced. So if glucose is the major product, it is difficult to determine how much glucose is produced inside the cell. However, it is easier to see how many oxygen molecules are evolving, what is the rate of oxygen evolution. So here if you were to determine the rate of photosynthesis, you check for how much oxygen is released. Okay, so on the y-axis you take how much oxygen is released, and on the x-axis you take the various wavelengths. So you will incident at this time not in a spectrophotometer, not for an individual pigment. You will incident the plant with monochromatic light, only 400 nanometer, 450 nanometer, 470 nanometer, and you will check for how much oxygen is released. Okay. So if you do that, you will get what is called an action spectrum. This is very similar to what we obtained when we discussed the Engelmann's experiment, right? Near the blue and the red wavelengths of light, all of these bacteria accumulated. Here you're seeing similar results, right? So when you compare the action spectrum with absorption spectrum, you will notice how for chlorophyll A, there are two peaks, one near the blue wavelength and one near the red wavelengths. And for chlorophyll A, this is for chlorophyll A, and for chlorophyll B, there's only one peak near the blue region. And for carotenoids, somewhere near the blue to green regions, you can find the peaks. But if you compare these pigments, absorption spectrum with this action spectrum, you will notice how wherever there are peaks for chlorophyll A, in similar regions there will be peak in the action spectrum too, which kind of indicates that chlorophyll A is the primary most directly involved pigment of photosynthesis. Okay, now why are so many pigments required? Why can't chlorophyll A alone do the job? See, the whole idea behind photosynthesis is to trap the energy present in sunlight and perform photosynthesis and produce glucose, right? If chlorophyll A alone is present, every pigment cannot absorb every wavelength. Every pigment can absorb only specific wavelength of light. Like that, if you have different pigments can absorb different wavelengths of light. Now you might ask me, what is the point of carotenes absorbing a certain wavelength of light if they are not directly involved in photosynthesis? What if they cannot directly perform photosynthesis? What if they are not directly involved in photosynthesis? Whatever energy it has absorbed, it can transfer that energy to chlorophyll, right? It need not directly be involved. But if you have different kinds of pigments, you can absorb different wavelengths of light. Chlorophyll, whatever wavelengths of light chlorophyll cannot absorb, some other pigments can utilize that wavelength, absorb the energy in that wavelength and pass on that energy to chlorophyll. That will make the process of photosynthesis a lot more efficient, right? So if you look here, from 400 to 700 nanometers is what the visible wavelength of light is. How much chlorophyll can absorb maximally? Absorption maxima is 650 to 700 nanometer. But you have 450 to 500 carotenoids, to 620 to 650 chlorophyll B. So what happens here? They will absorb the light, those particular wavelengths of light, and pass it on to chlorophyll A. Now you might ask me, what if chlorophyll A only ends up passing on its energy to chlorophyll B, which will be useless? Actually, chlorophyll A cannot pass its energy to chlorophyll B. I'll tell you, whenever energy is getting transferred from one pigment to another pigment, from higher energy to lower energy, it can get passed on. From lower wavelength to higher wavelength, it can get passed on. You've learnt in physics that wavelength and frequency are inversely proportional. Greater the frequency, smaller will be the wavelength. Greater the frequency, greater is the energy that that particular wavelength has, right? Even though carotenes are absorbing a lower wavelength of light, they have more energy, right? So that is why from carotenes to chlorophyll B, energy can pass. From here to here, from lower to higher, it will not spontaneously pass. Again, from chlorophyll B to chlorophyll A, it can pass energy, but not back from chlorophyll A to chlorophyll B. Because of this reason, all of these other extra accessory pigments that are there, they will help chlorophyll A by passing on whatever energy they've absorbed to make sure photosynthesis happens very efficiently, effectively, okay? Now, so within a photosystem that I drew like one dabba there, you're going to find hundreds of such pigments. Chlorophyll A will be at the center, and chlorophyll A is called the reaction center, and around the reaction center you have many pigments that are called antenna molecules or the accessory pigments. They are not directly involved, but of course they are very helpful. Also, if the sunlight is too harsh, sometimes it's too intense, it ends up damaging the pigments. So at such harsh sunlight, if chlorophyll A alone is present, it can get damaged. It can get damaged irreversibly, making sure that that particular plant cannot photosynthesize again, or that particular leaf cannot photosynthesize again. So by absorbing all of that extra energy, these other accessory pigments will kind of prevent the photooxidation of chlorophyll molecule and protect it, okay? So yeah, they are very beneficial, very good friends of chlorophyll A. And yeah, so these pigments along with certain proteins, certain enzymes and electron carriers is what forms a photosystem. Now there are two different types of photosystems, photosystem 1 and photosystem 2. We call it PS1 and PS2 in short. PS1 is called PS1 because it was first discovered. PS2 obviously was discovered later, so it's called PS2. Both of these are photosystems containing a reaction center, chlorophyll A, surrounded by many accessory pigments, and you also have certain proteins and enzymes, electron carriers associated with this. So that is the photosystem. At the center, reaction center, both of them have chlorophyll A. But there is a difference in that chlorophyll A. What is that difference? It's not molecular. Okay, so earlier I told you chlorophyll A and chlorophyll B are both chlorophyll molecules, but there is a slight difference in the R group. Here it's not like that. Molecular level, there is no difference. Both of them are identical chlorophyll A molecules. Then how are they different from each other? I'll tell you. PS1 has chlorophyll A in the reaction center that absorbs 700 nanometers absorption maxima it has. Okay, so that is called chlorophyll A 700. While the chlorophyll A in the reaction center of PS2 has an absorption maximum of 680, so we call that chlorophyll A 680. Now, how are they different from each other? Why are they absorbing, why are their absorption maxima different from each other if they're both the same at the molecular level, right? So, if that is your doubt, think of it like this. The energy level of this molecule, what wavelength it can absorb, depends on what are the proteins present around it, okay? So, the kind of proteins that are present around chlorophyll A 700 in PS1, the same proteins are not present around the reaction center chlorophyll A 680 of PS2. So because of the interaction of chlorophyll A with the surrounding proteins, different proteins have different amino acids have different side chains. So depending upon how this chlorophyll A molecule responds and reacts with the surrounding molecules, what wavelength of light it can absorb maximally, that will vary. It's more like if you're singing, right? If you're singing in a room. For example, let's say Naguvinane Sara ನೀನಾಗಿರುವೆ ನಡುಗಿದೆ ಕಂಗಳು ನೀ ಕಾಣದೆ. I know there was absolutely no necessity for me to sing at this moment because you're doing something important, but I just felt like singing. So what I'm trying to say here is, right now I sang the song in this room which has furniture, which has a computer, which has a dashboard and a lot of other things. I sang this song in this room and it sounded this way. If I sang the same song using the same voice without changing the pitch, the tone or anything in an empty room, right, in a large empty room, I would be singing the same way, but the voice does not sound the same, right? Because the acoustics of this room is different from the acoustics of that room. That does not mean I have changed, my voice has changed or my singing has changed. It just means that how my sound, my voice is interacting with the surroundings has changed. So this is very similar to that. So both of them are identical molecules, no change there. But how it is interacting with the surrounding proteins is what makes them have different absorption maxima. Now, let's talk about the locations. The location of PS1, you can find PS1 that I've represented in pink color. You can find it both in the grana and stroma lamellae. As you can see here, pink color is here and pink color is even here. So you can find them in the grana lamellae as well as the stroma lamellae, whereas the PS2 can be found only in grana lamellae. Orange color is found only here, right? So PS2 is represented by orange color. That can be found only here in the grana lamellae, whereas PS1 can be found in the grana lamellae as well as in the stroma lamellae. Okay. Now, let's talk about photophosphorylation. See, I told you we are discussing photosynthesis as a process in two distinct phases. The first phase is called the light reaction or the light dependent reaction. So, during light reaction, I mentioned earlier how ATP molecules form, right? So, the formation of ATP is called phosphorylation. Actually, phosphorylation in chemistry is a general term where a phosphate group is added to a substrate, you call it phosphorylation. But in the context of respiration and photosynthesis, when we discuss phosphorylation, we mostly talk about how ATP gets made. Okay, so when ATP is forming, what happens? Adenosine has two phosphates, another phosphate group gets added to it and it becomes adenose triphosphate, right? So addition of that phosphate group to ADP forming ATP, we consider phosphorylation. That is one type of phosphorylation. So in respiration photosynthesis, every time we say phosphorylation, we are talking about that. Okay. Now, here when ATP synthesis happens during the light reaction, the energy for phosphate group to get added to ADP. Remember, every time a new bond has to form, energy is required, right? So the energy required to add this phosphate group to ADP to make it ATP comes from where? The source of that energy is sunlight. So the energy from the sun is absorbed and through a series of redox reactions, electrons keep getting transferred from one electron carrier to the other. And when that energy during these redox reactions is utilized to synthesize ATP because it's happening in the presence of sunlight and because it's happening somehow, because of the sun's energy or the light energy, we call it photophosphorylation. Photophosphorylation is ATP getting synthesized in the presence of sunlight and this happens in photosynthesis only. Okay, so photophosphorylation can happen in two ways. I told you that ATP synthesis happens in light reaction, right? So that process, ATP synthesis during light reaction can happen in two ways. The first one is called non-cyclic photophosphorylation and the second one is called cyclic photophosphorylation. So we'll try and understand that. Now, here you're looking at the schematic representation of non-cyclic photophosphorylation. So what is this exactly here? You can see there's PS2 here and PS1 here. You should not ask me now why this is PS2, why is it not PS1, why is this PS1? Because this is first, this is second. The reason we're calling photosystems 1 and 2, like I told you earlier, is because of the order of their discovery. This was discovered first in the membranes. We had already given that PS1, the name PS1. This was discovered much later, so we have given it the name PS2. But if you look at the pathway of electron transfer, it happens from here to here. So that's why it's PS2 and PS1. And if PS2 and PS1 we're representing like this schematically, it's not like inside the chloroplast within the thylakoid membranes, there is like a Google map. It's not like the electron is moving straight and then going to one side and then going like that. That is not what this represents. This schematic representation is on a redox potential scale. Okay, so what is this redox potential scale? A redox potential is how easily it kind of measures how easily any molecule can let go of its electrons and get reduced, something like that. Okay, gets oxidized, not reduced, lose its electrons and get oxidized. So based on that, you have different values, and according to their values, different electron carriers are placed at different locations. Okay, so it's not like electrons have to go straight and then go right, then go left. That's not how it is. Actually, in the membrane, what happens is, if this is like your thylakoid membrane, let's say, so let's say this is your thylakoid membrane. So, oops, I took too thin. Okay, so let's say this is your thylakoid membrane. Within the thylakoid membrane, you will of course have the lumen, and inside the thylakoid membrane, you will have the lumen. And within the thylakoid membrane, you will have these photosystems. So within the membrane, you have PS2 and you have PS1 and you have a cytochrome B6F complex. So they are all located within the membrane. They are all within the membrane. Okay, and few other mobile electron carriers will be there. All of this is present inside the membrane itself. Okay, but here we are not showing that in the membrane to help you understand how electron transfer happens. So what you're looking at here is actually what happens in the membrane. Now when lights hit on the plant, okay, light hits on the plant, all of the leaves are illuminated. When leaves are illuminated, then all of the mesophyll cells will get light. Once the mesophyll cells get light, obviously it will hit on the chloroplast, and light will not be like, wait, first let me go illuminate PS2 and then let me go and illuminate PS1. Simultaneously, both of them are incident with sunlight. Though we start talking about it from PS2 to PS1, here it gets excited. It's not like until this comes here, this will not get excited. When sunlight hits both the photosystem simultaneously, you know, in chlorophyll molecule, you have a central magnesium, right? So from that, one electron will get excited. So from the chlorophyll A reaction center itself, electron will get excited. So electrons from both PS2 chlorophyll A, PS1's reaction center chlorophyll A, both of them will get excited. Okay, so that process is happening simultaneously. So the chlorophyll A680 loses the electron is first accepted by a molecule called pheophytin. Okay, so pheophytin is associated with the PS2 complex itself. Okay, so it's part of that complex. Right now I'm showing it somewhere away, somewhere distant, but actually in reality it is part of the complex itself. Some of them are parts of the complex, some of them are not part of the complex. Those details you don't need to know. For now, just understand that these are not very distantly present from each other. They are all located very close to each other. So based on the redox potential, they will lose or gain electrons. So this will absorb the sun's energy and electron will get excited. First, it is picked up by pheophytin. Pheophytin hands it over to plastoquinone. Plastoquinone will get reduced by accepting the electron. It will pass on the electron further to cytochrome B6F complex, which is located within the membrane, and then via plastocyanin, the electron will get transferred to the reaction center of PS1. Okay, so this is what has happened now. What about the fate of PS1? Electron got excited there. There are some molecules, intermediate electron carriers, which I have not written down here. It's not important for you. So let's say this is picked up by a molecule called ferredoxin, represented as FD in short, and that will pass on the electron to ferredoxin NADP reductase, which we call FNR. And as the name itself indicates, it reduces NADP. Okay, so this NADP reductase will accept the electron from FNR and it will get reduced to NADPH plus H plus. NADP is nicotinamide adenine dinucleotide phosphate. H just indicates that it is in its reduced form. Okay, so this happens. Now, this chlorophyll A lost an electron. This electron that was lost from PS2 kind of replaced the electron lost from PS1. It lost one, it got back one. So here it is stable, it can go through another round of excitation now that that lost electron has been replaced. What about PS2? It lost the electron has not yet come back. In order to replace the electron lost from PS2, very close to the PS2, you know, this towards the lumen side, there will be a water splitting complex present, and that will break or lyse water. Water is a very stable molecule, usually it does not, you know, split like that. But here during photosynthesis, photolysis of water happens. So after photolysis happens, protons, electrons and oxygen will be released. The electron from water splitting is used to replace the electron that is lost from chlorophyll A of PS2. This oxygen diffuses outside. When we say plants during photosynthesis give out oxygen, I told you earlier also it's coming from water and it is a product that is formed during the light reaction and not the dark reaction. Okay, so yeah, and these protons will stay inside the lumen itself. We'll talk more about it later. Remember that this photolysis of water is happening towards the lumen side, and this NADP reductase is present towards the stroma side. So the formation of NADPH is happening towards the stroma side. This happens towards the lumen, this happens towards the stroma. I will come back to that later. Now, here we can see another thing. I told you that photophosphorylation is all about making ATP, but so far we haven't spoken about ATP synthesis. ATP forms as electrons are going past the cytochrome B6F complex, or more so between plastoquinone and cytochrome B6F complex while electrons are being transported. ATP will get synthesized. How exactly it gets synthesized, I'll tell you as we move on. Okay, for now, this is all you need to know. In non-cyclic photophosphorylation, both of the photosystems are involved, which means that this has to happen in the grana lamellae only, because in the stroma lamellae, PS2 does not exist. In stroma lamellae, only PS1 is there. So the location for non-cyclic photophosphorylation is the grana lamellae. And another thing, I was about to say something, but I forgot. What happened? Yeah, now I remember. This is also called the Z scheme because it's like zigzag like that. It should have been called N, no, but it's called the Z scheme. This is non-cyclic photophosphorylation. Non-cyclic, why, if you're wondering? Because the electron lost from here does not cycle back to the same photosystem. Electron lost from here doesn't cycle back to the same photosystem. That's why it's called non-cyclic photophosphorylation. So in this process, we saw how using sun's energy, pigments trapped that energy, used it to synthesize ATP as well as NADPH, about which we've been talking since forever. And then water also splits, releasing oxygen. So this light reaction, one part is over. Now we'll move on and talk about cyclic photophosphorylation. Cyclic photophosphorylation can happen in two occasions. First, let's say only wavelengths of light beyond 680 nanometers is available to the plant. Okay, so it's not like the entire visible spectrum is available, but only wavelength beyond 680 is available. If only wavelength beyond 680 is available, chlorophyll A680 cannot absorb it. See, what happens is chlorophyll A680 can absorb wavelengths below 680 because that will carry more energy. Highest wavelength it can absorb is 680. Beyond that, energy will become less and chlorophyll A molecule cannot absorb that. So, if the plant is incident with light that is beyond 680 nanometers, then even though chlorophyll A680 exists, it will not be able to function. At that time, only PS1 will carry out phosphorylation. Secondly, it can happen in the stroma lamellae because in the stroma lamellae, PS2 is not even there. So, in stroma lamellae, only PS1 is there. That's why over there, cyclic photophosphorylation will take place. In cyclic photophosphorylation, you don't have both PS photosystems involved, only PS1 is involved. So, here you can see chlorophyll A700 is incident, illuminated with light. Central magnesium loses electron, captured or picked up by ferredoxin. Here we saw ferredoxin passes it on to ferredoxin NADP reductase, but near the stroma lamellae there is no NADP reductase, so it cannot pass it on to NADP reductase. What does it do instead? It passes it on to plastoquinone. See, it is going this direction. It's not going like this, it's going like this. So here ferredoxin will pass on the electron to plastoquinone and then further to cytochrome B6F complex and then further to plastocyanin, similar to what happened here, and then that electron will get passed on back to chlorophyll A700 from plastocyanin. Okay, so the electron that was lost by PS1 returns to PS1. So it's cycling back to PS1. That's why we call it cyclic photophosphorylation. Over here, similar to how previously we saw when electron is being transported from plastoquinone to cytochrome B6F complex, ATP synthesis happened, I told you. So ATP synthesis happens even over here. So if you look at the membrane, in the thylakoid membrane, if this is like the thylakoid membrane, inside the thylakoid membrane you have your lumen, and outside the thylakoid membrane you have the stroma. Okay, so within the thylakoid membrane you can see how PS2 is here, PS1 is here, cytochrome B6F1, B6F complex is here, and then you have plastoquinone here, plastocyanin here. All of the electron carriers will be present here, and as you can see, NADPH is forming towards the stroma, water splitting is happening towards the lumen. These are some things that I want you to keep in mind over here. Now, we have to talk about how exactly ATP synthesis happens during electron transport. Okay, so this can be explained by a hypothesis called chemiosmotic hypothesis. What is osmosis? Osmosis is a type of diffusion specifically of water molecules. Whenever water molecules are moving from a region of its higher concentration to a region of its lower concentration across a semi-permeable membrane, We call it osmosis, right? Why are we calling this chemiosmotic hypothesis? Here chemicals are, there is a gradient, concentration gradient of chemicals. That's why we call this chemiosmotic hypothesis. So here, this explains how ATP synthesis happens. Now, here there is a gradient that exists. How does osmosis happen? If there is a gradient between water concentration, then only water will move from one place to another. If they are both the same concentration, then there won't be any net movement of water molecules, right? Here also, a gradient has to exist first, and that gradient has to exist across a membrane. Okay, so here for ATP synthesis, you first need a membrane. Do we have a membrane? Yes, we have a membrane. It is a thylakoid membrane. And across the membrane, there should be a proton gradient. Is there a proton gradient here? Yes, there is a proton gradient. Within the stroma, you can find low protons, and within the lumen, the proton concentration is higher. There are three reasons which contribute to a higher proton concentration in the lumen. Let's talk about it. So first thing is, when water splitting happens, it happens towards the lumen. When water splits, electrons that are released are used to replace the electrons lost from PS2. Oxygen that is formed diffuses outside of the plant into the surrounding environment. What happens to the protons? The protons are accumulating right there. That is our first reason. What is the second reason? The second reason why there are more protons inside the lumen compared to the stroma is because when electrons are being transported from plastoquinone to cytochrome B6F complex, when this electron transport is happening, protons are pumped. Protons are pumped from the stroma side to the lumen side. Every time we say pump and transport, that means energy is utilized. That means transport is happening against a gradient. Already there are more protons present in the lumen, fewer protons present in the stroma. Despite that, more protons are forcefully being pumped into the lumen. So pumped, usually you've learnt about sodium potassium pump when it comes to nerve impulse generation and transmission. Over there, ATP is used for energy for pumping protons to wherever, okay, inside the membrane, what not protons there, sodium potassium ions, for them to be transported, ATP is used as an energy source. Over here also for pumping, energy is required. But for that energy, ATP is not used. Whatever energy is liberated as redox reactions are happening, see, look here. This is the redox potential scale. From high energy to low energy, when electron carriers are picking up electrons, some amount of energy will be lost, right? That energy is used for pumping protons from the stroma side into the lumen side. And this happens via the cytochrome B6F complex. So a proton pump is essential. So here we have a proton pump in the form of cytochrome B6F complex. Okay. So yeah, basically protons are pumped at that point. That also causes an increase in concentration of protons within the lumen compared to the stroma. And finally, already stroma has fewer protons compared to the lumen. But in the stroma is where NADPH is forming. For NADPH to form, NADP plus gets electrons from FNR. But where does it get protons from? It will pick up protons from the stroma. Already fewer protons there, but it is picking up even more protons from the stroma to form NADPH. That will further reduce the concentration of protons in the stroma. I want you to understand this here. 3 points are there. Here, because of water splitting, protons are accumulating in the stroma. Here, because of protons being pumped, it is getting accumulated in the lumen. I said stroma, right? It is in the lumen. It gets accumulated in the lumen. Third one is not protons entering the lumen. Third one is because protons existing in the stroma are getting used up and their concentration is further decreasing in the stroma. Okay, so these are the three reasons why a proton gradient develops. Okay, there is a proton gradient, fine, great. For ATP to get synthesized, there is an enzyme required that is called ATP synthase. So this ATP synthase molecule is a membrane bound enzyme. Okay, so this ATP synthase also is present within the membrane itself. This ATP synthase has two parts, a membrane bound CF0, Yeah, it's labeled here. A membrane-bound CF0 particle. It's like a transmembrane protein. That is embedded within the phospholipid bilayer. This pink color thing that you're looking at here, this is nothing but the phospholipid bilayer that you will find in the thylakoid membrane. Okay, so this entirely is that phospholipid bilayer, and here you have the CF0 particle. Now, for protons to get out from the lumen to the stroma, it uses this CF0 particle. It has like a tunnel of sorts through which the protons can move out. Now, I want you to understand, many people when they're reading NCERT, they get confused and they think the proton pump is this. This is not the proton pump, because for you to call it a proton pump, movement of protons have to happen against the gradient. Right now, what is happening? Right now is just diffusion. It is facilitated diffusion. There is more here, less here. From a region where protons are more to a region where protons are fewer in number, spontaneously protons are moving outside because they are charged particles and cannot directly pass through the phospholipid bilayer. This is just facilitating its movement. Okay, so as protons move through the CF0 particle, there is a part of this ATP synthase called the ATP synthase head, which we call the CF1 particle. So this one will change its conformation. This CF1 particle, actually it's like a peeled orange. It has many subunits. It will be holding on to one ADP molecule and one inorganic phosphate. As protons pass through it, move through it, it will combine this ADP and inorganic phosphate and produce ATP and release it to the stroma. Remember, we need ATP in the stroma, we need NADPH in the stroma. So it is towards the stroma that these products of light reaction are being released. Okay, in case you get confused, where is more protons, where is less protons, think about it like this. Where are the products of light reaction required? It's required for the dark reaction. Where is that happening? That's happening in the stroma. So ATP is released into the stroma, NADPH is forming inside the stroma so that they can be used up in the dark reaction. Okay, so this is what the chemiosmotic hypothesis is all about, and this explains how ATP is synthesized during the light reaction. Okay, now we have to talk about the dark reaction. Dark reaction, like I mentioned earlier, is also called the light independent reaction or the biosynthetic phase. Biosynthetic because here is when the actual synthesis of glucose will happen, and this does not directly depend on the light or sunlight, and it depends on the products of light reaction. So ATP and NADPH that are produced during light reaction will be used to drive the synthesis of glucose in the dark reaction. Now, the dark reaction is also called the Calvin cycle to honor the researcher, the scientist who elucidated the entire pathway. His name is Melvin Calvin. We talked about him a little while ago also. So what he did was he used radioactive C14. So C14 is an isotope of carbon that is radioactive. So in whatever carbon dioxide was exposed to the plant, or whatever carbon dioxide the plant was exposed to, same thing, no? Yeah. So that carbon was radio labeled. Okay, so it was C14. They will check at regular intervals within the plant tissue if there are any compounds containing C14. Okay, see, C14 was not originally available to the plant. You are providing C14 only in the form of carbon dioxide. But within the plant, if you find any other molecule that has C14, that kind of indicates that that molecule formed from C14 of carbon dioxide, right? So you will get to know the fate of carbon dioxide. What exactly is happening to carbon dioxide once it enters the plant cell? These are things that you can't look under the microscope and guess, right? So that's why these kind of studies are made. So he used radioactive C14 to study photosynthesis in algae, and it led to the discovery that the first carbon dioxide fixation product was a 3-carbon organic acid. It's called phosphoglyceric acid, and he worked out the complete biosynthetic pathway, and therefore it's called Calvin cycle. So the first product that we are talking about here, which has 3 carbons, is called phosphoglyceric acid, or in short PGA. To the 3rd carbon, phosphate group is added. That's why it's called 3-phosphoglyceric acid. Okay, now similar experiments, whatever Melvin Calvin, whatever experiments he performed using algae, similar experiments other people started to perform using different other plants. they noticed that this is not the only way through which photosynthesis happens or the dark reaction proceeds. In some plants, the first stable product did not contain 3 carbons. Instead, it contained a 4 carbon, it was a 4 carbon compound, which was called oxaloacetic acid. Okay. So they figured that it's not the same in every plant. In most plants, it is 3-PGA. In a few plants, it is oxaloacetic acid, the first stable compound. So based on whether the first product that is formed is oxaloacetic acid, a 4 carbon compound, or phosphoglyceric acid, a 3 carbon compound, you have two different pathways for carbon dioxide assimilation. If it is a C3 compound, it is C3 pathway. If it is C4 compound, it is called the C4 pathway. First, let's learn about C3 pathway, then we'll understand C4. So in C3 pathway, the first acceptor molecule of carbon dioxide was found to be a 5 carbon molecule called ribulose bisphosphate. Ribulose 1,5 bisphosphate. Ribulose is a 5 carbon containing sugar. It is a pentose sugar. It has two phosphates, one at the first one at the first carbon and one at the fifth carbon. So that's why it's called ribulose 1,5 bisphosphate. Okay. Now, why is it not called diphosphate like ATP? In ATP, what happens? Phosphate are attached end to end like that. You call it diphosphate. Here, they are two different carbons. You call it bisphosphate. Okay, first carbon and fifth carbon. Now, let's see what happens. Yeah. So, they figured that formation of glucose happens in a cyclic manner. And in this process, RUBP used to get regenerated. Okay, so what are the 3 steps we'll understand? We'll start off with RUBP. RUBP, like I mentioned earlier, is ribulose bisphosphate. Has it written? Yeah, it's mentioned here. RUBP is ribulose bisphosphate. To RUBP, in the first step, carbon dioxide from the atmosphere will get added. This process is called, this is the first step, and this process or this step is called carboxylation. So this carboxylation is brought about by an enzyme called Rubisco. Rubisco. It is short for ribulose bisphosphate, that is its substrate. Ribulose bisphosphate carboxylase oxygenase. Okay. Now, this Rubisco is called ribulose bisphosphate carboxylase oxygenase because Rubisco, let's say, is an enzyme like this. Okay. To one of its active sites, it will bind to RUBP. On to the other site, it can bind either to oxygen or to carbon dioxide. So, it can add carbon dioxide to RUBP or it can add oxygen to RUBP. In this case, it is showing its carboxylation activity. So, here it is showing carboxylation activity, but it can sometimes add oxygen also to RUBP. We will talk about that later. Since it has that ability, we call it carboxylase oxygenase. Like I told you earlier, the first stable product of photosynthesis is a 3 carbon compound, that is our 3-phosphoglyceric acid. Now, if you look at it, RUBP has 5 carbons, carbon dioxide has 1 carbon. We already know that 3-phosphoglyceric acid has 3 carbons. If there are a total of 6 carbons here, how is it that there is a 3 carbon compound formed? There will be a 6 carbon compound formed first. Because it is unstable, it will immediately split and give rise to 2 molecules of this 3 carbon compound. Since you have 2 such molecules here, you have a total of 6 carbons right here. Okay. Now, I told you that photosynthesis is a redox reaction where carbon dioxide will get reduced to form glucose, so reduction needs to happen. So in this step, reduction will happen. Ouch. Yeah. So the second step is reduction. During reduction, acid gets reduced to an aldehyde. Okay. But in your textbooks, they've not given phosphoglyceraldehyde. They've just mentioned triose phosphate. I will write the same thing here. So you will end up having a triose phosphate after reduction. And this contains 3 carbons. And there are 2 such molecules here. Each molecule gets reduced to 1 triose phosphate. Remember, I told you that during Calvin cycle, during photosynthesis, dark reaction, the products of light reaction will be utilized. It is at this step that the product of light reaction will get utilized. So, in this step, per molecule, you require 1 ATP and 1 NADPH. ATP will break down to produce ADP. I mean, it will get broken to form ADP and inorganic phosphate, and that energy is what will get utilized. And NADPH will get oxidized to NADP plus. Since there are 2 molecules here, this will require 2 ATP and 2 NADPH. This process is called reduction. Further, this triose phosphate is withdrawn from the cycle and used in the formation of glucose. Okay, glucose has 6 carbons. But here we have 2 such molecules. If glucose forms right here, RUBP will not get regenerated. If RUBP does not get regenerated, more glucose molecules cannot form. So this cannot directly be used for the formation of glucose. Of the 6 carbons here, 5 carbons will be utilized to regenerate RUBP. That step is called regeneration. For continuous operation of Calvin cycle, RUBP has to be regenerated, and that is our third step here. We call it regeneration. Okay. For the regeneration step, again, 1 ATP will be utilized. ATP, ADP. Now you don't ask me, here you wrote ATP on top, ADP down, here ATP down, ADP top, all of that does not matter, just look at the arrows. ATP is forming ADP means ATP is breaking and that energy is utilized. Here also ATP to ADP. Some books you might see ATP down, ADP up. What is up, what is down does not matter, the direction of the arrow is what will matter. Okay, so here you have regeneration. Now, of the 6 carbons that we have here, 3, 2 times is 6. Of the 6 carbons, if 5 carbons are given, utilized in the regeneration of RUBP, then we are left with only 1 carbon. But glucose has 6 carbons. So, in order for the formation of 1 glucose molecule, this Calvin cycle has to proceed 6 times. Okay? So, 6 cycles of Calvin cycle or 6 turns of Calvin cycle will help us get 1 glucose molecule. Okay, and this glucose can either be converted to or utilized in the synthesis of sucrose in case it has to be transported somewhere within the plant, or it can be stored in the form of starch. If we consider the same thing with respect to 6 molecules of RUBP, let's take 6 turns all at once. We're starting off with 6 molecules of RUBP. 6 times 5 is, here you have 30 carbons. Okay. Here you have 6 carbons. So you have a total of 36 carbons here. Here you already have 2 molecules. So 6 times, that is 12 molecules of a 3 carbon compound will give us 36 carbon, right? You have 36 carbons here. These 36 carbons will remain 36 carbons here too. Of these 36 carbons, 30 carbons are withdrawn. Okay, so from here to here, 10 molecules are used for regeneration. What is remaining? 3 molecules, no wait, 6 alva. 2 molecules will remain. Out of 12, 10 goes here. The remaining 2 is utilized here to form this 6 carbon glucose. Okay, so this is our C3 cycle or Calvin cycle. Yeah. So if you look at how many ATP and NADPH is utilized in the formation of one glucose molecule, you know, one glucose requires 6 carbon dioxide. You require 18 ATP. Why? 2 in the reduction step, 1 in the regeneration step per turn of Calvin cycle. So 3 ATPs per turn. For one glucose molecule, you require 6 turns. So 6 turns means 18 ATP, 6 turns means 12 NADPH, because 2 NADPH is required for one turn. A total of 6 turns will be 12 NADPH. In questions, if they have asked you for how many ATP, NADPH is required, how many carbon dioxide molecules are involved, just check in the question whether they are asking for one glucose molecule or per turn of Calvin cycle. Per turn means there will be one carbon dioxide molecule, 3 ATP molecules and 2 NADPH molecules. Okay. But if it's for one glucose molecule, these are your numbers. So that's about your dark reaction. Now, I told you that Rubisco is kind of choosy, it's kind of picky. It always in one reaction, in one active site it will bind to RUBP, but on the other active site it can either bind to oxygen or it can bind to carbon dioxide. It does not have like any special preference or anything. When they are both equal, like nearly equal in concentration, carbon dioxide and oxygen are both nearly equal in concentration, it has a greater affinity for carbon dioxide than oxygen. But when around Rubisco, there is more oxygen compared to carbon dioxide, it will choose oxygen, it will choose and bind to oxygen and it will add oxygen to RUBP. Okay, so it depends on the relative concentrations of carbon dioxide and oxygen. When carbon dioxide is more, it will pick up carbon dioxide. When both are equal, carbon dioxide, oxygen, both are equal, even then it has a preference for carbon dioxide. But when oxygen is much higher than carbon dioxide, it has no choice but to bind to oxygen. It's not like Rubisco is like, okay, carbon dioxide looks pretty, oxygen looks pretty, I should bind to oxygen. It's not something like that. It's based on the relative concentrations that is present around where Rubisco is there. Okay. So yeah, the binding of Rubisco depends on the relative concentration of oxygen and carbon dioxide. If there is more oxygen, it will show its oxygenation activity or the oxygenase activity. And if it has more carbon dioxide around it, then it will perform the carboxylation activity. So if it performs carboxylation activity, what's going to happen? Calvin cycle will proceed. But if it shows oxygenation, another process will happen that is called photorespiration. So when photorespiration happens, see, RUBP is oxygenated. RUBP has 5 carbons, oxygen has 0 carbon. You will end up, instead of getting 2 molecules of a 3 carbon compound, you will end up getting 1 molecule of a 3 carbon compound, 3-phosphoglyceric acid that contains 2 carbons, okay, and 3 carbons, and another molecule called phosphoglycolate that contains 2 carbons. So 5, 3 plus 2. Instead of getting 2 3-PGA, you're getting 1 2-carbon compound. And this 2-carbon compound is a little problematic. It is not good for the plant. It is kind of toxic, you can say. And in order to eliminate phosphoglycolate, the plant has to go through a process called photorespiration. Now what happens during respiration? Tell me. Oxygen is utilized, carbon dioxide is released. During photosynthesis, what happens? Carbon dioxide is utilized, oxygen is released. But here in this process called photorespiration, in the presence of sunlight, respiration like process will take place. Carbon dioxide that is already fixed will get eliminated, which is very unlike of photosynthesis. During photosynthesis, carbon dioxide should form glucose, but carbon dioxide should not get released. But during high light intensities, photorespiration will happen. I'll tell you why photorespiration happens and when it happens. Whenever the sunlight is too intense or very high level of transpiration is happening, plants will not be able to conserve water because a lot of water will be lost by transpiration. In order to conserve water as much as possible, plants will start closing their stomata. Okay, so when the plants close their stomata shut, carbon dioxide cannot enter the cells. Carbon dioxide cannot get into the leaf or the mesophyll cells. But oxygen also cannot escape outside. New carbon dioxide molecule is not getting inside. Oxygen is not getting out of the cell. So around the rubisco, more oxygen molecules will start getting accumulated. Okay, so when more oxygen gets accumulated, it will form phosphoglycolate. This is happening in the presence of high light intensities. It's a respiration like process because carbon dioxide is getting released. You call this photorespiration. So here when this happens, it's bad for the plant like I mentioned because phosphoglycolate has to be eliminated. When it is getting eliminated, ATP is utilized. Okay, so it's for a wasteful process, right? This was first of all not even essential. Now you're trying to eliminate it and for that you have to use ATP, which is a burden on the plant. Secondly, if this in this process of trying to eliminate or get rid of phosphoglycolate, you convert it into some other molecules, and during that process, fixed carbon dioxide is also released. Like carbon that is fixed is a part of organic compounds that is released as carbon dioxide. And in this process, unlike photosynthesis, glucose also will not form. If 3 molecules of phosphoglyceric acid forms, like in carboxylase activity of rubisco, at least glucose will form. Here, this is very opposite of photosynthesis because glucose is not forming here. ATP is used, but it's of no use because glucose is not forming. And whatever carbon is fixed with in organic molecules, that is also getting liberated. So overall, photorespiration is a bad process for the plant. It's a wasteful process for the plant energetically. Okay, so in order to escape or reduce the rate of photorespiration, different plants have evolved different adaptations. One such adaptation you can find in C4 plants. Wait, this slide was repeated. Yeah. In certain plants like maize and sorghum, monocot plants found in tropics, you find C4 pathway. C4 pathway has evolved to avoid or prevent photorespiratory losses. Okay, so in these plants, physiology is also different, anatomy is also different. The leaf anatomy is modified. In normal leaves, how do you find the leaf anatomy? You have the upper epidermis, lower epidermis, and then you have the mesophyll cells. And in the center, wherever veins are present, you will find xylem and phloem, that's the vascular bundles. And surrounding the vascular bundles, you will find bundle sheath. In most cases, it is the mesophyll cells where Calvin cycle will take place, lot of chloroplasts are present and photosynthesis actively happens. But in case of these plants where C4 pathway takes place, they have a different type of anatomy called Kranz anatomy. Kranz in German means wreath. What is wreath? It's like a ring, like structure that people put on their doors during Christmas. So that sort of thing. Why is it called Kranz anatomy? Kranz is not somebody's name here. Kranz is that ring or wreath sort of a thing. The reason is because in plants that show this kind of anatomy, the mesophyll cells surround the bundle sheath cells in the form of a ring. Centrally, you have xylem and phloem here. This is your vascular bundles. Surrounding the vascular bundles, you obviously have bundle sheath cells that I'm going to outline using yellow color here. Surrounding the bundle, vascular bundle is the bundle sheath. These are your bundle sheath cells. And surrounding the bundle sheath cells, you have mesophyll cells. These are your mesophyll cells. this entire ring. So bundle sheath cells and mesophyll cells are neighboring cells, right? So here, the way in which the bundle sheath cells and the mesophyll cells are arranged inside the leaf itself is different, because this is not something that we see usually, right? So arrangement wise, anatomy wise, they're different. If you look at bundle sheath cells, normally they do not contain chloroplasts, right? But in C4 plants, bundle sheath cells, okay, so these bundle sheath cells contain chloroplasts. That is unusual, number one. Secondly, those chloroplasts are agranule, which means within these chloroplasts, grana do not form. How does grana form? Thylakoids stack on top of each other and grana forms, right? But in these chloroplasts present inside the bundle sheath, agranule chloroplasts are there, meaning they are just like stroma lamellae. Okay, so that is the second thing. Thirdly, usually here chloroplast is not there. So Rubisco enzyme will not be there, but in this case bundle sheath contains high amount of Rubisco within the chloroplast. And normally in mesophyll cells, okay, where is bundle sheath? Here, yeah. Normally in mesophyll cells you will have high amount of Rubisco, but in this case mesophyll cells lack Rubisco. Mesophyll cells lack Rubisco. Instead, they contain an enzyme called peptase or pepcarboxylase. I'll tell you what is the reason for this. Why is photorespiration happening? Because relative to carbon dioxide, oxygen concentration is increased. How can you prevent photorespiration? If you make sure that surrounding Rubisco, carbon dioxide concentration is always maintained high, then photorespiration will never happen. They have this adaptation, they have evolved this way in order to make sure that the carbon dioxide concentration around wherever Rubisco is present is always high. Okay, so how do they accomplish that? I told you that only in the bundle sheath cells you have Rubisco. It's not there in mesophyll cell. Wherever Rubisco is there, only there Calvin cycle can operate. So in this case, in the leaves, mesophyll will not get involved in Calvin cycle. Calvin cycle occurs only in the bundle sheath cells. Okay, so how are the bundle sheath cells making sure that the carbon dioxide concentration there is always high? Firstly, they have chloroplasts. Chloroplasts are agranule. If they are agranule, grana are not forming, then PS2 will not exist there, right? If PS2 does not exist, then only PS1 is there. If only PS1 is there, then non-cyclic photophosphorylation cannot take place. Only cyclic photophosphorylation will take place. If cyclic photophosphorylation takes place, water splitting does not happen. If water does not split, oxygen does not evolve. That ensures that within the bundle sheath cells, oxygen concentration is maintained very low, carbon dioxide concentration is high. Also, in these bundle sheath cells, you can find high number of mitochondria, and those mitochondria, even if any oxygen is available, will utilize it for cellular respiration. So in that way, carbon dioxide concentration is maintained high at all times in the bundle sheath cells where Rubisco is active. Also, the cell wall of bundle sheath cells is very thick. So oxygen from surrounding cells can't randomly diffuse into the cell also. So that is how it ensures that carbon dioxide concentration is maintained high. Mesophyll cells lack Rubisco. Mesophyll cells will have chloroplasts, but they do not have Rubisco, so Calvin cycle does not operate there. Instead, another enzyme is present that is called peptase. What is its function? I will tell you now. Now, if you look at this, you can see how the bundle sheath cell and the mesophyll cells are neighboring cells. This is the bundle sheath cell, this is the mesophyll cell. Now, let's consider one mesophyll cell and one bundle sheath cell. Now that they are neighboring cells, you know that in between neighboring cells, plasmodesmatal connections will exist. So here is the plasmodesmatal connection or the cytoplasmic connection. If carbon dioxide has to enter the leaf, it has to enter through the epidermis. Stomata is present at the epidermis. So through the epidermis, it will enter the plant. First, it has to go to mesophyll, then only it will reach the bundle sheath, right? So first, stomata carbon dioxide either from here or here. First, it has to go through the mesophyll cells and then only it will reach the bundle sheath. So here you can see carbon dioxide present in the atmosphere enters the mesophyll cell first. Here is our mesophyll cell. This is our bundle sheath cell. Just a minute. This is mesophyll and this is bundle sheath. So you can see once carbon dioxide enters the bundle sheath cells, it will combine with water to form carbonic acid, and then it will dissociate, and then you have this bicarbonate ion that contains the carbon dioxide. This is carbon dioxide. Think of this as carbon dioxide. So this carbon dioxide contains one carbon. This combines with a 3-carbon compound called phosphoenol pyruvate. This itself is PEP, PEP. Okay. This enzyme is catalyzed by an enzyme called PEP carboxylase. So when a 1-carbon compound combines with a 3-carbon compound, you end up getting a 4-carbon compound, which is oxaloacetic acid. This is primary fixation, first fixation that is happening in the mesophyll cell. In mesophyll cell, rubisco does not exist, so this cannot undergo Calvin cycle. This cannot take part in the Calvin cycle. What happens here is, now this C4 oxaloacetic acid that has formed will either change into some other 4-carbon acid like malic acid or aspartic acid, and that acid, C4 acid, C4 meaning it contains 4 carbons. So that acid will get transported from the mesophyll to the bundle sheath via plasmodesmata. So the next step is transport. First primary fixation is over, then it's getting transported via plasmodesmata. Once it reaches the bundle sheath cell, it undergoes decarboxylation there. 1 carbon dioxide from the C4 acid gets removed. So you're taking out 1 carbon from a 4-carbon compound, a C3 acid will form as a result. Here you had 4 carbons, 1 carbon removed in the form of carbon dioxide, 3 carbons remain in the form of C3 acid. Now this C3 acid is transported via plasmodesmata back into the mesophyll cell. Over there, that C3 acid will regenerate to produce phosphoenol pyruvate. If you think about it, phosphoenol pyruvate is behaving like a vehicle. It's picking up carbon dioxide, coming into the bundle sheath cells, delivering it there, going back, pick up another molecule of carbon dioxide, come back in as a C4 acid, deliver the carbon dioxide, go back as a C3 acid, right? So that is what is happening. When this happens, you can ensure that at all times, wherever rubisco is active, which is in the bundle sheath cells, carbon dioxide concentration is maintained high. Okay, so this carbon dioxide is utilized by rubisco, and C3 cycle also happens here. C4 pathway is not independent of C3 cycle. If not for C3 cycle, glucose cannot get synthesized at all. So C3 cycle must happen. But it's not happening directly in the mesophyll cells like in C3 plants. Here, first primary fixation happens in the mesophyll cells. Later, again, fixation will happen very similar to how it happened in the C3 cycle over here in the bundle sheath cells. So C4 pathway includes the C3 cycle. They are not two entirely different things. Okay. So this is about your C4 pathway. So here is a table from your NCERT. We'll have to answer a couple of questions here. Cell type in which the Calvin cycle takes place. Calvin cycle takes place where in the C3 plants it takes place in the mesophyll cells. In C4 plants it happens in the bundle sheath cells. Cell type in which initial carboxylation reaction happens. In both cases it is in the mesophyll cells. How many cell types does the leaf have that can fix carbon dioxide? In C3 plants you have one type, in C4 plants you have two types, one in mesophyll, one in bundle sheath. Which is the primary carbon dioxide acceptor? In C3 plants it is RUBP, and in C4 plants it is phosphoenol pyruvate. Number of carbons present in the primary carbon dioxide acceptor. This one has 5 carbons, this one has 3 carbons. Which is the primary carbon dioxide fixation product here? Here it is phosphoglyceric acid, 3-PGA, and here it is oxaloacetic acid. Number of carbons in the primary carbon dioxide fixation product. Yeah, so this one has 3 carbons, this one has 4 carbons. Does the plant have rubisco? Of course, plants have rubisco. Where it has rubisco will change, but plants will have rubisco. If not for that, photosynthesis, dark reaction cannot take place. Next, does the plant have PEP carboxylase? C3 plants don't have, C4 plants have. Which cells in the plant have rubisco? In C3 plants, it is mesophyll cells. In C4 plants, it is bundle sheath cells. Carbon dioxide fixation rate under high light conditions. When the light is too intense, in C3 plants, photorespiration happens. So, carbon dioxide fixation kind of reduces. So, you can say low to medium in this case. In C4 plants, it does not matter because they anyway have a carbon dioxide concentrating mechanism in the bundle sheath. So, regardless of the light intensity, photosynthetic rate will be high there. Whether photorespiration is present at low light intensities. If low light intensity is there, in both cases, photorespiration will not happen. At high light intensities, whether photorespiration happens, In case of C3 plants, it does. In case of C4 plants, no, it doesn't. Whether photorespiration would be present at low carbon dioxide concentrations, yes. No, in C4. Whether photorespiration would be present at high carbon dioxide concentration. If there's high carbon dioxide concentration, it does not happen even C3 plants, right? For it to happen, relative to carbon dioxide, oxygen has to be very high. If carbon dioxide itself is high, even in C3 plants, it does not take place. What is the temperature optimum? C3 plants at high temperatures, because photorespiration happens and it's a wasteful process. Here temperature optimum is pretty low. It could be between 20 to 25 degrees Celsius, you can say. And in C4 plants, they have a much higher temperature optimum, let's say 30 to 40 degrees Celsius. Next, we have examples for C3 plants. Let's just write mango. And C4 plants, I've already told you, sorghum, maize, these monocot grasses. They show C4 pathway. And they have that Kranz anatomy. Okay. Now, factors affecting photosynthesis. We've come to the last part of this chapter. There are various internal factors as well as external factors that affect the rate of photosynthesis. The internal factors are controlled by the genes. For example, what is the number of size, age and orientation of leaves? This depends on the genes that a plant has. You can't expect a mango plant to produce leaves like a banana leaf, right? So it depends on the type of genes present in the plant, what will the shape of the leaves be, how expanded the leaf can grow into, what orientation the leaves will be. Will they be kind of horizontal, vertical like this, or will they be fully exposed to sunlight? How many leaves are present inside a plant? Age of the leaves, all of these are under genetic control. Number of mesophyll cells and chloroplasts, this also is under genetic control. Of course, if more mesophyll cells are there, more chloroplasts is present. Obviously, it's better for the plant. With respect to photosynthesis, the rate will be high. Internal carbon dioxide concentration and the amount of chlorophyll. Internal carbon dioxide concentration is basically in every one of our living cells, cellular respiration will keep taking place. As a byproduct of that, carbon dioxide will keep getting released, right? So that's the internal carbon dioxide concentration. More chlorophyll, more photosynthesis. And then we have external factors. External factors are mostly like the raw materials for photosynthesis. One is sunlight, availability of sunlight. Temperature, because all of these reactions that are happening during photosynthesis are controlled by enzymes. And you know, you've learnt about enzymes and how they function at different temperatures. In the chapter biomolecules, temperature affecting the enzymatic activity, you know, every enzyme has its own optimum temperature beyond which its activity will drop. Since photosynthesis has so many reactions and each step in each reaction is controlled by an enzyme, it will definitely be dependent on temperature. Carbon dioxide concentration, that is also one of the most important ingredients for photosynthesis, and water. All of these are external factors, right? And all of them will operate simultaneously. Simultaneously means at the same time. So, yeah. In order to understand how all of these will operate simultaneously, there's this one law called Blackman's law of limiting factors. We'll try and understand that. This is interesting. So what Blackman's law of limiting factors says is, whenever there is a chemical process, okay, so it became too green, let's use orange. Yeah. Whenever there is a chemical process taking place, in this case, the chemical process is photosynthesis. If that process is affected by more than one factor, in this case it definitely is affected by more than one factor. It is affected by light, it is affected by temperature, carbon dioxide concentration and even water, right? So it is affected by more than one factor. Its rate will be determined, the rate of the process. These are the factors. The rate of this process will be determined by that factor which is nearest to its minimal value. Now, what does that mean? Let's say, I'll give you one example. Whenever you're trying to make, okay, so let's say you're trying to manufacture a pen. This is the example that I commonly give. In a pen, you have different parts. You have the body of the pen, you have the refill inside, you have a cap, you probably have a grip. Okay, so there are 4 parts, let's say. You have the body of the pen, you have the cap, you have the refill inside, and then you have the grip. In this case, let's say in one factory, in one manufacturing unit, per second, the body will produce, the number of bodies that are produced is 100. The number of caps that are produced is 30. The number of refills that are produced is 70. And the number of grips that are produced is 40. Okay. So these can be thought of as different factors influencing photosynthesis. Now, if I ask you per second, how many complete pens can be assembled? How many complete pens can be assembled depends on the part that has the lowest value. In this case, cap is the least, 30 is the least. No matter 100 bodies are being produced, bodies of pen are being produced per second. But per second, you cannot get 10 completely assembled pens, right? You can only get 30 assembled pens because even though this is 100, you do not have enough caps to cover the pens with, right? So this will, this is what will determine the rate of the entire pens assembly. Now, let's say this is the lowest value. I increase this quantity to 50. Now, per second, can 50 pens be assembled? It can't, because now the lowest value is grip. We do not have sufficient quantities of grip. So, the highest number of pens you can assemble per second at this moment is 40. Now, if I increase 40 to, let's say 80, can I assemble 80 pens per second? I can't. Now, this became limiting again. Now, I increase this to 90. Can we get 90 pens per second? We can't. Now, this became limiting. Okay. So, even in photosynthesis, you will have different factors affecting the rate of photosynthesis. Whichever one is highly deviated from its optimum value, that is what will determine the rate of the photosynthesis. Now, whichever was the lowest, by increasing the quantity of the lowest component, I increase the overall rate of photosynthesis. Initially it was 30, but when I increased that to 40, overall rate also increased. But if I increased 100 to 110, overall rate would not have increased. So if you increase the factor that is closest to its minimal value, which is the least value, if you increase that only, you will get an overall increase in photosynthesis. Let's say the light is perfect, the temperature is optimum, water is great, but carbon dioxide concentration is not sufficient. If you go on increasing more light and adding more water to the soil, the plant will not photosynthesize better because carbon dioxide is not sufficient. So whichever one is the least, it's in its lowest value, suboptimal value, if you increase that, only then the overall rate of photosynthesis will increase. Okay, so it is this factor which will directly affect the process if its quality is changed. That's what it means. Sometimes light may be perfect, temperature is perfect, carbon dioxide concentration is perfect, water is not. So you add water. You add water, it is photosynthesizing well. Eventually the temperature will change as the day passes. Then temperature will become a limiting factor. A little later, the light will, it will become like time around sunset, like dusk time. Then light will become a limiting factor. So these are the things. Whichever one is near its minimal value, that is what will affect the overall rate of photosynthesis. Now let's talk about all of the different factors affecting the rate of photosynthesis, starting with light. There are three aspects of light that we must talk about. One is the quality of light, one is the intensity of light, and the other one is duration of exposure. Quality of light refers to the wavelength of light. We saw when we did that absorption spectrum graph that all wavelengths of light are not equally effective in photosynthesis. Plants prefer certain wavelengths of light at which photosynthetic rate will be high. We saw that in action spectrum as well, right? So here, if you're constantly giving a plant only green wavelength of light, it's obviously not going to photosynthesize because chlorophyll cannot absorb green. That's why it's imparting green and we see it as green color, right? So it depends on what wavelength of light is available to the plant. Secondly, light intensity. Light intensity can be thought of if you have like an area, let's say 1 centimeter square of area. Per second, how many photons hit that area is the light intensity, or 1 meter square, whatever you take, whatever area. How many photons will be incident on that area per second will give intensity, light intensity. You may have noticed after sunrise, light is there during the day, early morning light is there, even during afternoon. But if you go out and stand in the sun, you will feel that burning in the afternoon which you don't feel during early morning, right? The intensity is very high as we proceed from early morning to midday or afternoon, you say, and then again it will progressively decrease. Throughout the day, the light intensity will not be the same. So the intensity of light also is important. Next, duration of exposure to light. Of course, you might feel like, oh, sunlight is such an important part for photosynthesis. If we provide light to plants 24/7, then it might photosynthesize very well. It's not really true. Plants also during the day, because of sunlight can cause cancers in us, skin cancer in us. Like that, excessive sunlight can damage the pigments in plants and certain molecules can get damaged within the plant. Only during those dark periods during the night is when all of those molecules will get repaired. You may have heard how all of the damage that we do to our body during the day will get repaired during the night when we go to bed, right? It's very similar even for plants. So in fact, every organism on our planet, including plants and animals, we have evolved keeping in mind that sun comes during the day, he goes back during night, right? So keeping that in mind, keeping that diurnal pattern in mind is how all living organisms are evolved. With respect to photosynthesis, you might feel like plants don't need a dark period, but plants do need a dark period in order to reset and de-cleanse, what do you call it, detoxify and all of that. So too much exposure can damage pigments irreversibly. I told you about that even earlier, right? So the duration of exposure is important. Next, if you look at the graph where you have light intensity on x-axis and the rate of photosynthesis on y-axis, with increase in light intensity, initially you can see an increase in photosynthesis, but after one point, it will not increase any further. Even if you increase the light intensity, it could damage the pigments, or at that point, some other factor can become limiting. Okay, so linear relationship exists between incident light and carbon dioxide fixation rates at low light intensities. At higher light intensities, gradually the rate does not further show any increase as other factors will become limiting. Light may be perfect at that point, but temperature could become a problem. So one other factor becomes limiting. So light saturation occurs at 10% full sunlight. Okay. So in most cases, light is not a limiting factor for plants unless the plant is growing in a dense forest where there are huge trees that are forming a canopy of sorts, and the floor of the forest is very shady, and the vegetation or the plants that are growing on the surface may not be able to receive sufficient sunlight. Even those plants will be adapted. Plants that are adapted to such shady light conditions will grow there. Okay, so that is the thing. And yeah, beyond a point, if you keep increasing the light intensity and the duration of light also, if it's too long, then chlorophyll will start getting broken down. Other pigments also will start getting damaged. Okay. Eventually, it will cause a decrease in photosynthesis. Now, let's talk about carbon dioxide concentration. This is the major limiting factor for photosynthesis. In our atmosphere, from where plants take in carbon dioxide concentration is about 0.03 to 0.04%. If you increase that even slightly up to 0.05%, you will see an increase in the rate of photosynthesis. Beyond that, in Kannada we say ati adre amritanu visha anta. That means too much of elixir is also like poison. Too much of anything will turn bad. Even if it's like the nicest thing, too much of anything will be bad. Okay, so carbon dioxide also, if it increases beyond a limit, it can be damaging to the plant. Yeah, and if you talk about C3 and C4 plants, they respond differently to increase in carbon dioxide concentration. If you see at low light intensities, light intensity itself is low. If you increase the carbon dioxide concentration, photosynthetic rate will not increase because, okay, you're giving carbon dioxide to the plant, but for Calvin cycle to happen, ATP is required. ATP plants get from light reaction, but light intensity is not strong enough. So even though carbon dioxide is there, for fixation, ATP and NADPH are not sufficient. So it will not show any increase in the photosynthetic rate. But at high light intensities, C3 and C4 plants, both of them will show an increase in photosynthesis. Now speaking of C3 plants, they have a higher saturation level compared to C4 plants because C4 plants anyway have their inbuilt carbon dioxide concentration mechanism in bundle sheath cells, which C3 plants lack. So for C3 plants, you need more carbon dioxide for saturation. So C3 plants show saturation only beyond 450 microliters per liter, whereas in C4 plants, it is much lesser, around 360 microliters per liter. So currently, whatever carbon dioxide is available to the plants, this is limiting for C3 plants only. C4 plants are such geniuses, they have a way to ensure around rubisco always carbon dioxide is present. Okay, now temperature. Temperature, I told you, is important because all of the photosynthetic reactions are controlled by enzymes, and enzymes have their own optimum temperatures, right? So, though even in light reaction, enzymes are involved, the enzymes of the dark reaction is what will get mostly affected. They are the ones that are mostly temperature controlled. So, if too high temperatures are there, C4 plants can still survive somehow, but C3 plants, photorespiration will start, and they will not be able to photosynthesize very well. That's why they have a lower temperature optimum compared to C4 plants. Okay, so even at higher temperatures, they show high rate of photosynthesis, C4 plants, because of their bundle sheath carbon dioxide concentrating mechanism. But C3 plants, since they don't have that, they have a much lower temperature optimum. The temperature optimum depends on the habitat too. Like for example, if plants are growing in a country like India, which is a tropical country, it will have a higher temperature optimum. If it's growing in Greenland, Iceland, Scotland, then the temperature optimum will be low. According to what kind of environment it's growing in, what is the ambient temperature there, it will also have its own temperature optimum range. Okay, now moving on to the last part, water. Water is an important ingredient for photosynthesis, right? It's a requirement. It is a substrate for photosynthesis, but it has indirect effect on photosynthesis. Whenever water is less in a plant, the plants, to conserve whatever water is available, they will start closing their stomata. That will limit the entry of carbon dioxide into the plant. That way Calvin cycle cannot proceed. Secondly, if the stomata closes shut, photorespiration can happen. That is a wasteful process, waste of energy. Secondly, thirdly, in order to limit the surface area exposed to sunlight and control transpiration even further, plant leaves might wilt. Instead of being exposed like that, they might droop down like that. When that happens, the exposure to sunlight is limited, so light reaction cannot happen properly. So that way, indirectly, it will affect the rate of photosynthesis. So even though it's one of the reactants of light reaction, the effect of water as a factor more is through its effect on the plant rather than directly on photosynthesis. Water stress causes stomata to close, hence reducing carbon dioxide availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. That's what I just said, right? Okay. So with that, we've come to the end of one of the most dreaded plant physiology chapters, photosynthesis in higher plants. Such a beautiful process it is. I love it. Okay. So I hope you love it too moving forward. And we should all say thanks to plants, because if not for them, there's no food, there's no oxygen. We exist because of them, right? So yeah, that's about it. I'll see you in the next class with another chapter. Until then, take care of yourselves, stay healthy, stay happy, keep learning, study hard. Bye bye. Thank you.